Hi Truth Risers, welcome, and as always I hope you're doing great. In this video we're going to look at some retrodictions. So what are retrodictions anyway? A retrodiction is an explanation of past events that are inferred from the laws that govern those events. As pertains to evolution then, a retrodiction is an observation that indicates the truth of evolution and common ancestry, but isn't something that would have been a prediction necessarily. Instead, it's something that is discovered in hindsight to be consistent with or support evolution and common ancestry. Retrodiction 1. Vestigial organs, for example, the vestigial hind limb bones of the gray whale. It's clear that whales don't have legs sticking out of their bodies. They certainly don't have hind legs protruding out of their backside, but they do have, as part of their skeleton, tucked inside of their body, small hind limb bones. These are very, very tiny little bones within the whale that have persisted in their anatomy and are non-functional. It's very hard to explain why these vestigial hind limb bones are so small and have persisted, except in the context of common ancestry and evolution. Retrodiction 2, vestigial genes. Vestigial genes are genes that we have in our genome and that other organisms also have in their genome. But in our genome, they don't work. They're non-functional. They cannot produce the target product that other organisms can produce that have this particular gene in their genome. Let's look at a few examples of this. Mammals have a gene for making yolk proteins, but mammals do not produce actual egg yolk. Instead, they produce an empty sac, but the sac doesn't contain the yolk. Even though the sac doesn't contain it, the gene for making it, the yolk protein, is still in the genome of mammals. Another example is production of vitamin C. We have the gene for making vitamin C in our genome, and so do other primates, but we can't produce vitamin C because the gene doesn't work. Most other mammals, on the other hand, can produce their own vitamin C. We have to get vitamin C from outside sources that we consume in our diet. Our bodies simply don't make it because the gene for making vitamin C, though we have it, doesn't work. Most other mammals also have the gene and can make vitamin C. Why would we all have the same gene for making vitamin C and see that it is functional and produces vitamin C for some organisms but not for others? This is consistent with evolution in that it indicates common origins, different genomes, same genes. But there's been a change that occurs, an evolutionary change, which results in a cessation of vitamin C production for our species and other primate species. And the last vestigial gene that I'll mention is the gene for making olfactory receptor proteins. We, along with other mammals, have genes for making olfactory proteins, which are very important for our sense of smell. Although we have olfactory receptor genes in our genome, and we share the presence of these olfactory receptor genes with other species, many of ours are non-functional or inactive. Yet these genes are common to many animals. Some animals, such as mice, elephants, and dogs, have more functional genes for olfactory receptors than humans. Actually, dogs have about 3 times more than we do. This is a retrodiction that has been consistently demonstrated in evolution, and there are other examples of retrodictions as well. So these are 3 examples of genes that we have in common with other organisms that work in some organisms but are no longer functional for us. I should mention that these examples are courtesy of Dr. Muhammad Noor, who is an evolutionary biologist at Duke University. We'll discuss one more, the retrodiction related to biogeography and what it tells us about the evolution of organisms, in a future video. Do you have any retrodictions you'd like to share? If you do, please drop them in the comments. We'd love to hear from you. And thanks so much for watching. Take care.